Today, I would like to talk to you a little bit about this, what I consider to be a fairly remarkable process, and that is the process of grafting. When we cut plants and we join different plants and different species together, and they have this ability to grow as one. Now, grafting itself has been practiced for an extremely long time. So we can go back over 1,000 years and we see examples of successful grafting, whether we go back to ancient China or ancient Greece and ancient Rome. Now, grafting itself is very widely used. This is just some examples that I've taken within Europe, but there's major applied interest in grafting, and it's estimated that there's over 2 billion plants per year are being grafted, and likely the real number is much, much higher than this. And examples include many of our common vegetables, such as tomatoes and melons and watermelons, are being regularly grafted. But we also see examples with our fruit trees, and also it's becoming more and more common now in forestry to graft. So this is a major economic contributor. Now, my group is really focused on trying to understand the fundamental mechanism for how plants are able to successfully graft. We want to know how plants can successfully graft so they're compatible, and also why certain graft combinations fail. We want to take this information to then improve grafting. And we also want to look at examples of natural grafting. So this could be things like root fusion that occurs underground when plants grow in close proximity. We also see examples of branches fusing together. And we also have this really interesting example of parasitic plant infection. This is an example of a mistletoe growing on an apple tree. And again, looking at this example of plant-plant interactions and whether we can use this information to somehow try to better understand grafting and to improve grafting. Now, I'm not the first person to work on grafting. There's been a long history of successful graft research. And people in the 1970s and 1980s defined a series of steps that were absolutely critical for the formation of the graft. The first step had to be the adhesion of the tissues, so they had to stick together, followed by the formation of callus, the stem cell-like tissue, and finally you had the formation of the vascular connections. If we go back 100 years ago, people were studying things such as geraniums here. So we can see an example of a geranium graft cross-section that was drawn here over 100 years ago. And of course, nowadays we have modern tools and techniques that we can use to study graft formation. My group has really pioneered and done a lot of research using Arabidopsis micrografting. And with very young Arabidopsis tissues, we are able to graft them through the hypocotyl here in the middle, and we get very high success rates. And this allows us also to graft many plants per hour. And using these type of techniques, we've described genes, mechanisms, and pathways that are important for the successful formation of the graft. We developed time courses, so we basically described when the tissue is attached, followed by the reconnection of the phloem, followed by root growth, followed by the connection of the water conducting tissue or the xylem. And we could do assays such as this one here, where we look at the transport dynamics of fluorescent molecules, such as carboxyfluorescein or GFP. Now, today I'm going to talk to you about a couple different stories that my lab has been doing. I won't initially focus that much on Arabidopsis, but I would like to present two examples where we used micrografting, where I think we've contributed both fundamental and applied interest. So the first is our work with monocots. Now, monocots, of course, are an incredibly important group of plants, and they were previously thought to be ungraftable. And this was thought to be due to a combination of their absence of vascular stem cells, the vascular cambium, and the fact they also had scattered vascular bundles, which perhaps made alignment of the two halves of the plant more challenging. In my own grafts with stems of maize, they were a disaster. But what I and also our collaborators in Cambridge realized was that if you used incredibly young tissues, so you're talking about 1 day old seedlings, and you dissected out the shoots of the embryo, and then you replace this with another seedling, you actually got fairly good success rates. So here you can see an example where we perform this technique on rice grafts, and we're getting success rates approaching probably 60 or 70%. And we get nice fusion of the tissues, enabling a successful graft between two different genotypes of the rice. Our collaborators in Cambridge also extended some of these observations and were able to successfully graft palm trees and also banana plants using these micrografting techniques. What was very surprising, though, is that when we did these grafts, Normally, grafting has to be within very closely related species or within the same genus. But there was something very special about monocots. Suddenly, we were able to graft very distantly related plants to each other. We were able to graft rice to pearl millet, rice to sorghum, and other different combinations here. The success rates were not extremely high, but considering how distantly related these species were, it was still very surprising that even after weeks or months after grafting, they were still viable. Now, another example I'd like to present is the work that we've been doing with gymnosperms. So this is another major group of plants. And traditionally, gymnosperms have been grafted in horticulture. And in Sweden, there's approximately 20,000 trees are grafted a year using these type of methods on more mature trees. And we realized that micrografting was probably going to be a much more efficient way to do this. So we developed techniques to micrograft very, very young seedlings. We do it primarily in vitro, but here you can see some examples here where we've actually grafted the trees growing in soil with a little collar. And using these methods, we had very high success rates, and we also had grafting rates that were 4 times faster than traditional grafting. Now, using these types of grafting techniques, we were then interested in comparing how do gymnosperms compare to dicots in terms of their grafting dynamics. We did transcriptome sequencing of both Norway spruce and also from our previous work in Arabidopsis thaliana, and we observed actually a remarkable conservation in the process, not just at the physiological level in terms of things like phloem and xylem reconnection, but we could actually see genes and homologs, such as those associated with cell cycle. You can see some in Norway spruce here, the homolog in Arabidopsis, and they're both being upregulated at very similar times during the grafting process, both in the scion, or the top part of the graft, but also in the rootstock. And we could also observe this for wound response genes like WOX13, phloem-related genes like APL or APL. And we also discovered another gene that hadn't been previously associated with regeneration or with grafting, PAT1. And we actually found this in our Norway spruce data sets. We looked at our Arabidopsis data set. We saw conservation of this. And then we actually took the PAT1 homolog from Norway spruce. We transformed it into Arabidopsis, and we were able to partly rescue the Arabidopsis mutant phenotype during grafting assays, suggesting not only was there temporal conservation of gene expression dynamics, but there's actually functional complementation of this transcription factor. And finally, when we did these experiments, we were also able to take these plants and let them grow to maturity. So these trees are approximately 3 years old. And we were able to graft pine trees, Pinus sylvestris to itself. We could also graft pine trees within the genus. So in this case, we grafted Pinus sylvestris to Pinus contorta. But remarkably, using micrografting, we were able to graft 2 different genuses together. So the Pinus, or the pines, were able to successfully graft to the spruce trees, or the Picea. And this is a graft combination that would normally never be successful using traditional grafting techniques, but micrografting allowed this combination to succeed. And now 5 years later, these plants are still viable, suggesting a long-term compatibility. So I'd like to move on to another example here where we actually look at how plants respond to development and to their environment. So I like to think of grafting as a really nice example of developmental plasticity, how plants can change their development in response to wounding, and in this case, the grafting. And what you can see here is that when we actually graft a plant, we get the reconnection of the vasculature here in the middle, but we can also get 2 outcomes that are quite common. One is you have successful graft healing in the middle, and the other is you get the formation of this structure called an adventitious root or a de novo root. We were quite interested to try to understand why this is occurring. There's certainly a competency across the hypocaudal to form these 2 types of tissues, but we wanted to investigate this in more depth. And rather than using the Arabidopsis hypocaudal, we actually decided to use the leaf. And what happens here is that you can take a leaf and you can wound the petiole or the stem here, and it has 2 outcomes. In some cases, our leaf petiole will actually form roots here. And in other cases, it can form this callus-like tissue, which we think is also very important for the formation of the graft. And we really wanted to try to understand the process and the mechanism that was actually regulating these 2 developmental outcomes. And to make a long story short, what we realized was it was actually the availability of water at the wound site that seemed to regulate this. And the way that we did this, we used a couple different assays, but one way is that we could actually change the percentage of agar these plants were regenerating on. So when we regenerated cut plants on a very low percentage of agar, so the water availability is very high, these plants regenerated primarily roots. When we start to increase the percent of agar, so we go up to 2% agar or 3% agar, the water availability becomes very, very low. And in this case, the wound site now transitions from forming roots to now forming this wound-induced callus, this type of stem cell. We could take this further. We tested this with tomato tissues, and we saw something very similar. When water availability was high, the cut tissue regenerated roots. When water availability was low, the cut tissue regenerated wound-induced callus. Now, there's a technique in horticulture called air layering. And this is simply wounding a stem and then putting soil against the wound site to then induce root formation. And we were very interested in taking air layering. And in this case, we could either apply wet soil to the cut and we saw roots forming, but we could also apply dry soil to the cut. And in this case, we had callus then forming the cut and healing it. So we think there's actually a lot of endogenous environmental relevance for the plant. Depending where the plant gets wounded, it needs to decide whether it's going to form roots, if it's in a very damp environment, such as the soil, or if it gets wounded in a stem, it needs to then cover and seal that tissue. There's probably also some biotechnological interest. So of course, promoting root formation in environments like root-inducing media is very relevant. So if you're doing your root-inducing media on a very high percent agar, such as 1 or 1%, you might have very low efficiency of forming roots. And if you can reduce your concentration of agar, then your efficiency of root formation will become much higher. Okay, so that was all good and fine, but we really wanted to try to understand the mechanistic basis of this. And for this, we actually started to investigate auxin response because we thought auxin was very important for the formation of roots, the work that many others before us have actually done. And we looked at an auxin responsive marker called DR5. And what we realized was that under high water conditions, or low water conditions, we induced this DR5 responsive, auxin responsive marker. But what was surprising wasn't the magnitude of expression, was actually the location of expression. So under high water conditions, we could actually see this increase or this maximum of auxin response above the cut site. And under low water conditions, this auxin maxima became much, much lower. And you can see here 2 days after wounding, it became even more pronounced, and then 4 days later. So it looked like it wasn't necessarily the fact that just auxin was activating during the cutting process, but it's where auxin maxima were occurring that was critical. So we want to know why this was, and we looked a little bit more deeply at 2 other stress-related hormones. One was ethylene and the other was jasmonic acid. And in both cases, we could see them induced under high water conditions. So we could see our ethylene, our transcriptome, an ethylene responsive gene, ACS6, another ethylene gene activated within 3 hours of wounding. And we had some jasmonic acid related genes also activating. And you could see here with our reporter, with high water conditions, we get much, much higher response of this reporter than under low water conditions. And very similar for our jasmonic acid. It has much higher response in high water than low water conditions. Okay, so we have some stress hormones are being induced by the high water availability. Is this actually influencing auxin? And that's what we saw. So what we realized is that when we take intermediate water conditions that form both callus and de novo root regeneration, we treat it with an ethylene precursor or with methyl jasmonate, and we can actually then transition this to root fate. We can also look at our auxin reporter. We can treat our auxin reporter either with our methyl jasmonate hormone or with our ACC precursor. And we basically, instead of having a more uniform auxin response that you might see with intermediate water conditions, we can actually move this auxin response away from the cut site to then promote the formation of roots. So to make a fairly complex story short in this case here, what I would like to try to argue is that upon wounding, plants are monitoring the external environment to see water availability. And depending on this, they can then induce the formation of roots at the cut site, or they can form wound-induced callus at the cut site. And we think this has direct relevance for grafting. So under high water conditions, you very typically will get adventitious roots. This is one of the first pieces of advice that I'll give people when they're using our in vitro grafting techniques is to really reduce the water level. When we have low water, we get things like wound-induced callus formation forming, which we think is very important for grafting success. And here you can see a successful graft when we have low water. Okay, so for the next part of the talk, what I would like to do is get into a second story here that my group has been very interested in, and this is more broadly talking about plant-plant interactions. And we've discussed for the last 15 minutes about plant grafting, but there's another phenomenon that I think is also developmentally very interesting, and that is the process of plant parasitism. In parasitic plants, there's approximately 4,000 species that are known in the world, and many are important pathogens, and they also form interspecies connections. And in the case of parasites, they have a remarkable ability to infect hosts that are often completely unrelated to them or very, very distantly related. And you can see here, this is a parasite infecting its host and forming a xylem bridge connection between them, which in some ways analogously looks a little bit similar to the connections of xylem here that form between the scion and the rootstock. Now, our favorite species to work on is a member of the Orobanchi. It's called Therospermum japonicum. So it's actually closely related to a lot of the economically important parasites, such as Striga or Orobanchi. And it's nice because it infects lots of different species, including Arabidopsis thaliana. So it allows us to form a lot of interesting and important experiments using the tools and resources of Arabidopsis. And you can see here's PJ growing in the presence of Arabidopsis. It's a root parasite and it forms an infective structure known as the haustoria here, right where my pointer is. And if you look closely, this is where it forms a xylem bridge, and it can then use that to withdraw nutrients from the host Arabidopsis species. Okay, so we realized fairly early on we wanted to look for parallels between grafting and parasitism to see if we could use one process to help understand the other. And in both cases, we realized there's a very strong induction of cytokinin response. So we have a cytokinin responsive reporter here during grafting, ARR5, and we have the same reporter being activated in our host plant, Arabidopsis, and also in our parasite, Therospermum. So the parasite has another cytokinin responsive reporter, TCSN, and it's activating in purple, and Arabidopsis is activating in green. And we wanted to know what the biological relevance of this was. So in collaboration with colleagues both in the north of Sweden in Umeå and also in Japan, we basically investigated this using hormone profiling. And we realized that both in the parasite and host, we have a massive increase in cytokinin levels upon infection. And you can see that here in our Columbia infected, it's much higher than uninfected. And we can also see that in our Therospermum infected has a much higher levels than uninfected plants. So what's the biological relevance of this? And we realized that actually during the process of plant infection, you can see this nice stem swelling that's occurring in the host. So mistletoe infects its apple and causes swelling. PJ, Therospermum, infects Arabidopsis and also causes this tissue swelling. And it's been known for quite a while that actually radial growth is influenced by cytokinin levels. So what we did is we repeated some of these assays with cytokinin receptor mutants in the Arabidopsis host, and this totally blocked our tissue expansion or hypertrophy. We also repeated this in an Arabidopsis biosynthesis mutant of cytokinin, and it still occurred, really suggesting that actually the cytokinins were coming from the parasite, moving to the host, and causing the stem swelling. But what was very interesting is this stem swelling, we didn't really see it in the parasite. So the parasite was resistant somehow to this hypertrophy. It was really happening in the host. So what was happening? And we treated the parasite with cytokinins, and we had a very surprising result. And that was the parasites basically no longer infected their hosts when they were treated with exogenous cytokinins. So we could treat them with things like BA or kinetin or transietin. We could treat them with an inhibitor of cytokinin receptors, PI55, and this then actually caused an increase in haustoria numbers. We could generate transgenic parasitic plants using a cytokinin degrading enzyme called cytokinin oxidase 3. This actually lowers the amount of cytokinin in the parasite, and this led to more infections occurring, more Haustoria numbers. So really suggesting that cytokinins were actually somehow having a repressive effect in the parasite. So what we had here is a situation where cytokinins are induced during infection. They're having a repressive effect in the parasite, but they're actually moving into the host to cause this stem swelling or hypertrophy. And this actually reminded us a little bit of the situation in other symbiosis systems, such as autoregulation of nodulation, where the nodules are producing factors that can then move systemically to actually suppress the formation of further nodules. So we decided to test this using something called a split root system. We took a 1-month-old Therospermum plant, we divided it into 2 halves, and we could then infect this on either one half of the roots, the other half, and we could either do it at the same time, or we could infect first on one side of the plant, wait many days, and infect on the other side. And what we observed was that if we do the infection on the same half of the plant, or one half at day 0, the other half at day 0, we get the same number of Haustoria. But then as we start to delay the second half infecting, so 3 days, 5 days, 7 or 10 days, we get much fewer Haustoria happening. So the infections that were occurring at day 0 were inhibiting the ability for more infections to form at day 10. And our final experiment was here to basically investigate endogenously whether this system was indeed driven by cytokinin, so this auto-regulation system. And for that, we went back to our cytokinin oxidase overexpressing plants, and under non-transgenic plants, at day 10, again, you see this suppressive effect versus day 0. We get more Haustoria at day 0, and if we wait 10 days, the other half of the plant produces much less. In a cytokinin oxidase expressing plant, much fewer cytokinins. We actually get more Haustoria forming at day 0, but we lose this suppressive effect at day 10. So what we think is happening is that established Haustoria, or these infection sites, are producing cytokinins, which can then move systemically through the parasitic plant to then inform it about the number of Haustoria that it's forming and to actually shut down and to regulate its Haustoria numbers. So with that, I think I'm coming to the end of my time and my talk. And I would just like to say that, my group has really tried to pioneer the interactions or the study of these plant-plant interactions, focusing a lot initially on grafting. And our work in species like Arabidopsis, Gymnosperms, and monocots, I think has provided a nice framework for how plants successfully graft and also broaden the range of species that we can graft. We've used Arabidopsis, and we think the discoveries made there can be very much applicable to other species. We're also very much interested in parasitic plants and how they're actually able to both form these infective structures and to regulate them. And finally, our future goals are things like identifying markers of incompatibility. So how can we actually cause more plants to successfully graft? And can we actually investigate more using young tissues for successful grafting? And with that, I'd like to thank the people in the group who did all the work, including my funders and collaborators. And with that, I'm happy to take questions.
Thank you, Dr. Melnick, for that fascinating presentation. And our committee members, please feel free to unmute yourselves.
Okay, so I'll.
Okay, yeah, Dr.
Wow, so go ahead.
Mhm. Mhm. Oh, sorry. Okay. Professor Ralph Bock, please go ahead. Yeah.
Yeah, thank you very much for a great talk. So I have 2 questions actually. In your parasitic plant system, the Therospermum is a hemiparasite, right? So it conducts photosynthesis, and I was wondering whether you've tried to manipulate the source sink relations in the system, for example, by using a herbicide to block photosynthesis in the host, and then determine how this impacts the interaction between the parasite and the host, the cytokinin flow, and these other things that you're looking at.
Yeah, I think it's a relevant point. I mean, it's many parasitic plants are extremely strong sinks, but like you say, the hemiparasites actually, and especially Therospermum, it's a facultative parasite, so it can actually live on its own quite happily, and it seems to shift its transitions to parasitism depending on its environmental conditions. So we haven't done a lot of work to try to turn our Therospermum into slightly more perhaps, changing it into more obligate or even making the host to change its conditions. What we have done is to try to at least cut off the host to block its sort of source of sugars and its strength. And it looks like, I mean, the parasite is still very much interested. As long as the host is alive, it's going to go for it. And it looks like what Therospermum is looking for is specifically nitrogen. It's not interested in the sugars, it wants nitrogen.
Mhm. And if I can ask another question, in the monocot system that you got to work, so what do you think is the key anatomical feature in the very, very young seedlings that actually makes it work? Is it really just the more meristematic nature of the tissue, or is there also some other anatomical aspect to it, like arrangement of the bundles or, something else?
Yeah, so I think it's actually related to the first point you said. So I think it's related to the stem cellness of the tissues. So, most monocots don't have vascular cambium, so they can't divide their mature tissues, but they actually still, in principle, should have procambium. So when we have these very, very young seedlings that haven't fully differentiated their vascular tissues, I mean, at one day, they still, in principle, probably don't have fully differentiated. We can graft them and they still have an ability to basically divide these procambial-like tissues and actually form the connection between them. And what we've noticed is that if we wait even 5, 6, or 10 days after germination, the efficiency of grafting almost drops dramatically. So it can go from 80% to 10%. So it really needs to be these, I consider them undifferentiated or only partly differentiated cells.
Great. Thank you.
Thank you. And maybe Professor Luan?
Okay, so in the grafting, the JA and ethylene are promoting factors, right? How about ABA? The ABA is associated with less water. And you're talking about the J and ethylene probably related to more water or high water status. So
Yeah.
ABA.
Yeah, so that's really interesting. And when we did our transcriptome, actually ABA, so this is basically related to this fate decision. So under high water conditions, we're producing a lot of these roots, and that's probably mediated by ethylene and jasmonic acid. Under low water conditions, we're getting a lot of ABA responsive genes. So we think ABA is playing a major role there, and that would make a lot of sense. When we did exogenous ABA treatments and we also tested ABA mutants, I mean, basically, we see a complicated situation. And I wonder if this has to do with sort of crosstalk with ABA and sugar signaling, or there's something that we're basically messing up. We don't have the right threshold. We're blocking both processes somehow. So it wasn't as clear cut from a genetic point of view. But at least the transcriptome was very, very clear. So I think there's something interesting going on, but we haven't been able to dissect it further.
Thank you.
Okay. Any other questions?
Okay, Charles, during the grafting, what availability does what availability cause the oxygen level change or cause hypoxia situation, induce hypoxia signaling? Have you checked that?
Yeah, so I mean that would be really interesting actually to investigate further. And at the moment we haven't done anything with that, but I think you're absolutely right. I think this need for hypoxia and regeneration and stem cellness is really interesting. So it would definitely be worth us investigating further.
Okay, thank you.
Okay. Okay. Thank you for the great discussion. And once again, thank you, Dr. Malnick.
Thank you.
Our next speaker is Dr. Li Wan from CEMPS, Chinese Academy of Sciences. His research focuses on plant immunity and crop design with broad spectrum resistance, particularly on the mechanism of on the mechanism of immune receptor activation and ion channel function. Here we are present enzymatic and ion channel activities in plant intracellular immune receptors. Dr. Wang, please go ahead.
Okay, thanks, Wenrong, for the introduction. So, hello, everyone. I'm Li Wang from Chinese Academy of Sciences, Center for Excellence in Molecular Plant Sciences. First of all, I want to thank Molecular Plant for giving me this opportunity to participate in this event and present my research. Today, I will talk about enzymatic and ion channel activities of plant intracellular immune receptors. Plant pathogens and pests are responsible for up to 30% of crop loss globally. Understanding plant immunity for improved resistance breeding strategy represents the most eco-friendly way of solving this problem. Plants rely on immune receptors to detect pathogens and activate defense. Plant cell surface immune receptors perceive conserved microbial features to trigger basal defense known as PAM-trigger immunity, PTI. Plant intracellular immune receptors known as resistant protein or NR recognize specific pathogen factors to induce effector trigger immunity, ETI. ETI is a strong defense and cause localized cell death at site of infection to restrict pathogen spread. ETI confirms effective immunity against pathogens and is critical for plant resistance. Plant NR genes are important sources for resistant breeding in agriculture. Typical NR contain 3 domains. The C-terminal L domain is responsible for effector recognition. The central NB domain functions as a molecular switch controlling activation of the whole protein by oligomerization. The N-terminal singular domain is either a tier domain or CC domain. And we are mainly interested in the tier subclass of NRs. Tier NR functions as a sensor of pathogen effectors to initiate immune signaling. Downstream of sensor tier NR, a special class of NR with a typical CC domain, known as CCR domain, functions like helpers to execute immune functions. There are two classes of helper NRs, ADR1 and NRG1. 3 lipase-like proteins, EDS1, SEG1, and PAD4 function in this pathway by transducing immune signals from sensor tier NR to helper CCR NR. In terms of function, the EDS1 PAD4 ADR1 branch controls resistance. The EDS1 SEG1 NRG1 branch controls cell death. In 2019, when I was postdoc in Jeff Danker's lab, we discovered the tier domain of tier NL function as NAD cleaving enzymes to produce small singular molecules. In 2021, we found that both CCR, NR, ADR1, and NRG1 function as a calcium channel. Following studies trying to understand how the enzymatic activity triggers the calcium channel activity. In the same year, Jianming and Jijie found that the famous CCNL ZA1 resistance zone functions as a calcium channel. In 2022, 2 breakthrough studies led by Jijie and Jian identified two types of singular molecules. One type is PRIP-AMP-ADP that binds to the pre-existing EDS1 PAD4 heterodimer and induce its association with ADR1. The other type is ADP and di-ADPR that binds to the pre-existing EDS1 SEG1 heterodimer and induce its association with NRG1. In 2019, when I was postdoc in Jeff Danker's lab, we first discovered the tier NADase activity, and we found that the plant tiers cleaves NAD into 2 paths, LAM and ADPR. The nuclear molecule ADPR can cyclize and give rise to cyclic ADPR, CADPR. But we found that plant tier domains produce a CADPR isomer instead of canonical CADPR, which means that ADPR cyclizes at a different position. At that time, we did not know the identity of this CADPR isomer. In 2022, Boston Kobe's group resolved 2 CADPR isomer structures, 2' CADPR and 3' CADPR. And then we know that plant tier domains mainly produce 2' CADPR. And some plant tiers also produce small amount of 3' CDPR. To investigate function of 2 CDPR isomers in plant immunity, we chose the 4 bacterial proteins, AB tier and HOPY that produce 2' CDPR, AA tier and HOPA1 that produce 3' CDPR. We found that AB tier and HOPY specifically induce the heterotrimer formation of EDS1, PET4, ADL1, but not EDS1, SEG11, or NRG1. AA and HOPA1 can do neither job, while plant RPS4 tier can do both jobs. And also AB tier and HOPY specifically activate the function of EDS1, PET4, ADL1 when overexpressed in benzimiana, depending on their enzymatic activity. These results point to a link between 2' CDPR and EDS1, PET4, ADL1 function. Using mass spectrometry, we found that plant or bacterial tier induced EDS1, PET4, ADL1 heterotrimer proteins do not contain any 2' CDPR, but PRIP, AMP, or ADP. Cryo-EM structures analysis confirm PRIP, AMP, ADP as the OKI molecule. Close inspection of the chemical structures of these small molecules shows that hydrolysis of pyrophosphate bond in 2' CDPR will give rise to PRIP, AMP. And indeed, after incubation of 2' CDPR with Arabidopsis protein extract, we could detect the production of PRIP, AMP. Direct application of 2' CDPR, but not 3' CDPR on Arabidopsis leaves induce the expression of defense marker gene PR1. The induction is dependent on EDS1, PET4, AD1, but not on SEG11 or NRG1. Transcriptomic analysis of 2' CDR treated co-zero leaves revealed 2,500 upregulated genes involved in various defense pathways, and 35 downregulated genes, mainly involved in photosynthesis and oxygen signaling, while mimicking activation of effector immunity. Moreover, 2' CDPR enhances the resistance of Arabidopsis against Pseudomonas syringae. To summarize our findings, we demonstrate the immune function of 2' CDPR by hydrolytic conversion into PRIP that induce the heterotrimer formation of EDS1, PET4, AD1 to activate defense signaling. Zhu Hua's work also demonstrate that in rice is PRIP that induce the heterotrimer formation of EDS1, PET4, AD1. So importantly, given the functional conservation of PRIP and the EDS1, PET4, AD1 module in all plant species, 2' CDPR bypasses the recognition specificity of plant tier NR immune receptors against the pathogens and offers a new strategy to activate broad spectrum resistance. Then what about 3' CDPR? Even though we do not know the function of 3' CDPR in plant immunity, in this collaborative study with Magnesium Mura, we show that bacteria immune proteins produce 3' as a single molecule to activate antiviral immunity, and some plant tier domains produce 3' as a minor product and so can cross-activate bacterial immunity. Tier domain has conserved immune function in plants, bacteria, and animals. Its enzymatic activity needs to be well regulated to maintain homeostasis. In this study with Yang Zhao, we identified and conserved serine residues in tier domains, cross kingdoms that can be phosphorylated, and phosphorylation inhibits tier enzymatic activity. In plants, it's the calcium-dependent protein kinase CPK that phosphorylates tier signaling to balance growth and immunity. And this model summarizes our understanding of tier NR signaling pathway. The remaining gap is how the two helper NR, ADR1 and NRG1, are activated to be oligomeric calcium channel at the plasma membrane, and what is the relationship between the oligomer and the heterotrimer. To investigate the activation mechanism of ADR1 and NRG1, we first look into the plasma membrane association of ADR1 and NRG1. And found that helper NRs use a special mechanism to localize at the plasma membrane. Sequence alignment in the N-terminal domain, CCR domain of ADR1 and NRG1, and the CC domain of another helper called NRC from Arabidopsis and benzimiana identified 2 positive charged residues. One is in the alpha-2 helix and one is in the alpha-4 helix. the two positive charge residues are not conserved in general CCNRs. Taking ADR1 as example, we found that mutating the positive charge residues and labeling residues in either alpha-2 helix or alpha-4 helix affected phosphorylated binding in vitro. Combination of the two had a more obvious effect and completely lost binding towards phospholipid, especially PI4P. PI4P is the major contributor to the active electrostatic potential of plant plasma membrane. Mutating both alpha-2 helix and alpha-4 helix almost turned ADR1 into a cytoplasmic protein. Upon activation by pathogen effectors, TLNR produce small molecules ADPR, ADP, and di-ADPR to induce the heterotrimer formation of EDS1, SEG11, NRG1, and organization of NRG1. To further dissect this process, we use two different approaches to activate TLNR signaling pathway in Benzimiana, certified as promoter-driven expression of factors of Q, which represents strong activation due to overexpression of the effector, or bacteria PFO to deliver effector ZOP-Q, which represents a lateral way of effector secretion. And ZOP-Q is recognized by endogenous TLNR in Benzimiana. By co-IP, we found that weak activation preferentially induced the heterotrimer formation of EDS1, SEG11, NH1A. By blue native gel, we found that strong activation preferentially induced organization of NH1A. And this study suggests that EDS1, SEG1, NH1A heterotrimer likely represents the intermediate state before NH1 organization. And previously, we identified NH1A loss of organization mutant L134E. And in this co-IP, we show that NH1A L134E stabilizes EDS1, SEG11, NH1A heterotrimer formation, further indicating that TLNR produces more molecules, triggers heterotrimer formation, but may not be sufficient to trigger NH1 organization. Consistently, a study from Jonathan Jones and Jen's group demonstrated that PAM-triggered immune PTI is required for NH1 organization. And taking the advantage of NH1A L134E mutant that stabilizes the EDS1, SEG11, NH1A heterotrimer formation, we are able to obtain the EDS1, SEG11, NH1 L134E heterotrimer protein complex for radium structure analysis, while wild-type NH1A did not work. Even though we use full-length proteins for structural analysis, the full-length NH1A L134E with only one single mutation, but we are only able to observe part of the NH1A, the C-terminal WHT plus LR, and the N-terminal part is not observed in structure, indicating that the N-terminal part is disordered and not involved in the interaction with EDS1, SEG11. And we further showed that the interaction interfaces between EDS1, SEG11, and NH1A are critical for NH1 organization and cell death function. And there are three NH1 paralogs in apoptosis. NH1A and 1B are full-length proteins that positively regulate cell death with redundancy, while NH1C is a truncated protein that Xin Li showed as an active regulator of cell death when overexpressed. Interestingly, NH1C contains exactly the WHT plus LR region of NH1A that is responsible for interaction with EDS1 SEG11, indicating that Arabidopsis evolved such fragment of NH1C with core interacting domains of NH1A to sequester EDS1 SEG11. We first obtained the cryo-EM structure of EDS1, SEG11, and NH1C, and found that NH1C develops further interactions with EDS1, SEG11 to outcompete NH1A. So NH1C is a strong interactor to sequester EDS1, SEG11 and actively regulates the functional EDS1, SEG11, NH1 heterotrimer and the cell death function. Jie and Jen also showed that NH1C is a higher affinity binder for active regulation. And we show that HAP-NR, including NH1A, rely on positive charge residues in the CCR domain for phosphorylated binding and plasmembrane association. The N-terminally truncated NH1C has no CCR domain, so we look at subcellular localization of NH1C and found that it indeed has low longer localized to the plasmembrane. But in the cytoplasm and nucleus, in the PNAS paper, we also showed that nuclear cytoplasmic distribution of EDS-171 is critical for cell death function. To further test if NH1C coincides with EDS-171 in cytoplasm and the nucleus to achieve more efficient sequestration of EDS-171. We've used a myris toylation site to the N-terminals of NH1C to force its localization as a plasmembrane, and found that NH1C, once localized as the plasmembrane, lost its ability to inhibit NH1A trigger cell death. So in this case, we think Epilox evolved such a truncated protein, NH1C, that maintains core interacting domains of NH1A, loses plasmembrane association, and takes advantage of its nuclear cytoplasmic distribution for efficient sequestration of EDS-171 and elective regulation. Our study revealed some key singular events of how the enzymatic activity of tRNR activates the calcium channel activity of CCRNR, including both positive regulations and negative regulation. But there's still some important questions to be answered in this pathway. For example, we propose that the two heterotrimers likely function as a intermediate state. So do they have a function in regulating plant immunity? Our study suggests that EDS-171 NH1 likely has a transcription activation role in the nucleus, and also how the transition from the heterotrimer to oligomer occurred, how PTI is involved in the activation of NH1 and ADR1. And also, if ADR1 and NH1 function as calcium channels to trigger resistant cell death, and what are the calcium decoders downstream of ADR1 and NH1 to specifically regulate cell death and resistance. And also, the whole TR pathway is complicated, and how this whole pathway is lactically regulated to maintain homeostasis is also unknown and needs further clarification. And with that, I want to applaud the people involved in the work, some of the students and postdocs in our lab. And also we collaborated with Yu Zhang on the structure work, so helped us a lot on the crime structure analysis. And also we collaborated with Yang Zhao on the tier phosphorylation work, and we worked with Magnesium Mura on the bacteria immune signaling work, and also the funding agencies. So, and thank you all for listening, and I'm happy to take questions. Thanks.
Thank you, Dr. Wang, for your excellent talk. And now we welcome questions from our committee members. Please feel free to unmute yourselves.
You know, I don't have raising the hand button. I don't know where to raise the hand, but if I may, maybe I'll ask a question for Lee. Yeah, so do you think the CPK regulation of TIR, right, regulation, it's actually inhibition, right? Inhibition of a TIR enzyme activity, right?
Yes.
Do you think it serves as a feedback mechanism or some kind of a crosstalk mechanism?
we think it's likely a feedback and a buffering mechanism to attenuate TIR activity, because when immunity is activated, that's also always calcium signaling, right? The calcium
Sure.
likely activates BK and to attenuate TIR signaling, and especially the TIR-only proteins. And TIR-only proteins has been shown to be involved in the basal defense and the PAM, even PAM trigger immunity. So maybe that's likely how the CPK regulates TIRs to attenuate or buffering immunity.
Yeah, the reason I ask that is because, calcium is also involved in a lot of abiotic stress processes, right?
Yes.
And
Yes.
there's some kind of crosstalk between abiotic and biotic response.
Yeah.
That's why I thought, well,
Yeah. Actually,
Mm.
this work originated from Yang Zhao's work on the TNR SNP1's involvement in the osmotic stress. So osmotic stress activates SNP1 and has found CPK inhibits SNP1 to maintain plant growth during osmotic stress.
Mhm. That makes sense. That makes sense. Xiaofeng?
Hi, Wanli. Very nice talk. So I have one question. You mentioned that TLR can degrade NAD to produce the small molecule. So during this reaction, do you think the TLR can also affect the redox balance, or even affect the production of the ROS directly or indirectly?
Oh, we don't have any evidence to show this small molecule has activation role or has anything to do with ROS. We think it really affects treatment to parasitic with the Arabidopsis leaves really trigger like ATI-like response. So maybe some systematic resistance as well, but
So,
we don't see any ROS effect. So
okay, because NED and NEDH is one of the redox regulation system. So I wondering maybe the tail NLR can directly affect the redox balance in the cell. So maybe indirectly affect the ROS production and eventually cause the cell death. Is this possible?
uh, we don't really think so, because
Mhm.
let me talk this way. Okay, so the EDS1 so 2' CDPR is hydrolyzed into PRMP to activate the EDS1 PAD481 pathway, and this pathway is run for resistance. It's not really much involved in cell death, in Arabidopsis at least. So from my understanding, I think it's not likely that it will has a role in to contribute to ROS and cell death. So.
Okay. Another related question is, what's the true biochemical functions of the EDS1, PD4 of this family? I remember a long time ago, someone predicted that EDS1 PD4 may be a lipase. Is this true? Do you have
Oh,
any idea
EDS1,
about their
PAD481, they are
common
lipase.
functions?
They are lipase-like proteins.
Mhm.
Yes, that's been and has very conserved active site. But so far, I think some genetic studies from Jen showed that the lipase-like activities of EDS1 and PAD4, they are not required for resistance when they made the transgenic lines, the complementation lines. But I think we actually did some work. Is we mutate the lipase-like activity of EDS1, the benzene EDS1. We complement the Bansi EDS1 mutant with wild type or the EDS1 lipase-like catalytic dead mutant. We found the NB EDS1 catalytic dead mutant is weaker in cell death activity. So we think it might have some role in regulating cell death, but so far it looks like in resistance is not really required. So.
Okay, thank you.
Okay, thank you. Professor Fankhauser, please. Thank you.
Thank you very much for this nice presentation. I also have a question about these NAD breakdown or products. The first one is, can you actually measure the level of these different, 2' CADPR, 3' CADPR, and PRIB? Because, how do they change during the infection process? Can you measure them in the plant?
For 2' CADPR, we can. It's a stable molecule, but not for PRIB and P, as you can see here. So we incubate the 2' CADPR molecule with Arabidopsis protein extract, and after override, we can detect the production of PRIB and P. To detect PRIB and P, we need to add in purified EDS1 pet4 81 heterodimer proteins to protect it, and then denature the protein and do mass spec to detect PRIB and P. So PRIB and P is very highly unstable molecule, as we know in vivo for sure, and also in vitro is not that stable. But 2' CADPR is a stable molecule that we can try to quantify in vivo, I think, because.
But it hasn't been done. Nobody has seen whether the levels change during the infection process, or.
Okay, because. Actually, something I need to mention is that's actually the tier-only proteins, like the Arabidopsis RBA1, it produces a lot more prime CADPR than the tiers of tier NR. And in this 2019 science paper, so we can use the effector to HBA1 to activate the tier-only immune receptor RBA1, and then that's like activation of ETI. So then we try to detect 2' CADPR accumulation. There is accumulation of 2' CADPR during ETI, mediated by tier-only immune receptors. So we think it's functioning in some cases for the quantification of these 2' CADPR changes. But the series is not really that's because TNR does not produce that much, because we think TNR has the ability to produce, to make PRMP directly. But 2' CADPR is definitely a precursor of PRMP. And actually, that's an enzyme that's involved in conversion of 2' CADPR to PRMP, and we know the enzyme already.
Okay, I was going to ask, what is doing this catalysis? Because the reason I was asking is the level of these small compounds, the rate limiting step to lead to the production of these complexes? Or is it something else, which is the rate limiting element or the regulated element in the assembly of these complexes?
Yeah, I think even there are many factors. We think even PRMP, I mean, it's highly unstable in vivo. There's definitely enzymes involved in the breakdown of PRMP and the homeostasis of this molecule as well, because they are danger molecules, right? They directly activate immunity, and this PRMP and the single molecules, the PRMP ADP and the ADPR ADP di-ADPR, they're both direct activators. So these molecules has to be well regulated in vivo in homeostasis. And we have identified some. We have actually identified some enzymes involved in the homeostasis of these molecules in collaboration with GG. Yes.
Thank you.
Okay. Thank you for the engaging discussion, and thank you again, Dr. Wang.
Okay.
Our third speaker is Dr. Bing Wang from the IGDB, Chinese Academy of Sciences. Her research explores how strigolactone regulates plant architecture and rhizosphere interactions, with a focus on nutrient responses, shoot branching, and parasitic resistance. The talk is titled Strigolactones from Architecture to Rhizosphere. Dr. Wang, we look forward to your talk.
Yeah, thank you for the introduction and thank you for the invitation. It's my great honor to introduce my work about strigolactone from architecture to rhizosphere. I'm from the Chinese Academy of Science, IGDB. Yeah. As we all know that plant hormone is important for the crop yield and stress tolerance. There are 10 groups of plant hormones, and they are involved in the green revolution, seed size, fruit ripening, and disease resistance, drought resistance, and insect resistance. And how about strigolactone? It is initially discovered as a small molecule to trigger the seed germination of striga. Striga is an important devastating disease in the world. And then in 2008, it is identified and as a plant hormones. And the most important function is to repress shoot branching. Then people found that these hormones have important functions in the shoot development, root development, and leaf development. And importantly, it has different functions from other hormones is that it can separate it from root to the rhizosphere and can mediate the interaction with both the striga or banshee and also the AM fungi. That's to say it is important for both parasitism and symbiosis. And so why we are interested in this hormone? Because architecture is very important for crop production. And the architecture trees is mainly including the tiller number, that is shoot number, plant height, tiller angle, and the panicle size. For the tiller number regulation, especially in cereal crops, the strigolactone is the most important hormones to determine this trait. So we first focus on the biosynthesis of this hormone. And yesterday, Professor Yan Li has given a very fantastic talk about strigolactone biosynthesis. We also found
Sorry,
that.
Dr. Wang. Excuse me, Dr. Wang. Sorry, I'm sorry to disrupt, but the screen didn't move. Would you like to check your screen? Sorry.
How now?
No, it still didn't work. Yeah. Can you move it?
Move to the next page, yeah.
Maybe I move it.
Sure, it can.
Yes. Can you?
Maybe full screen?
This one?
No? Yeah, this work. Yeah.
Okay. Oh, okay.
Yeah.
Okay. The strigolactone is the endogenous level is very low, and it is the lowest hormone among all the discovered hormones. So it is hard to research, but we found that the low phosphorus can trigger the biosynthesis of strigolactone to more than 600-fold in rice. So what's the mechanism? We found that two important transcription factors, ASP1 and ASP2, which is also important for the symbiosis, is responsible for the induction. If we mutate these two transcription factors, the induction is blocked. And then we found that the triggered strigolactone biosynthesis can regulate the shoot development and root development, that is the architecture features, and also can regulate the nitrogen absorption and the phosphorus absorption. And to help the plants to adapt to the low phosphorus condition. If we increase the expression of these two transcription factors, we can improve the biomass and green yield of rice seedlings, rice plants, and growing in the low phosphorus and the medium phosphorus field. So that is important for agriculture. And then we move to another question is how does the hormone is perceived, because although the receptor have been identified in 2009, but the perception mechanism remain controversial. And they are based on the structure analysis, scientists have identified two complexes. The first one is the D14 receptor complex with the F-box protein D3 and ASK1. In this complex, scientists found that upon the signal perception, there is a conformational change of the receptor. Then the hormone is hydrolyzed to a small molecule called D-ring and to trigger the signal transduction. But another group found that they think that the D14 can interact with the D3 CTH motif, and this conformation they think is important for the signal transduction. And in this complex, the hormone do not hydrolyze, so they are controversial models. We want to know which conformation or which complex is important for the signal perception in plants. And so we analyze the conserved amino acid of the receptor and found that there are 165 conserved amino acids, and we mutate it one by one to evaluate its function in the signal transduction. We found that about 11% of the conserved amino acids is required for the interaction with D3. And interestingly, this amino acid is localized in the lead region, and this region undergoes the conformational change, and also many important residues is in the interaction phase of the D14, D3, ASK1, the complex. And the important amino acids covered most of the reported mutants in rice, Rhodopsis, pea, petunia. That is, say this screen is very useful. Then we perform the genetics and the biochemical assays to evaluate their roles in vivo. We found that if we we mutate 3 amino acids in the D14 and the D3 full-length complex, and as this one, and the receptor function is blocked. That is to say, it has no roles in each important signaling events of the pathway. So that is to say, the D14, D3, ASK1 complex is important for the signal perception. How about another complex? We mutate an important amino acid. In these mutant proteins, the complex cannot form. Surprisingly, we found that this protein it works very well in plants. That is to say, it is functional receptor, and it can even elevate signal perception. And we do another genetic analysis, that to say we overexpress the D3 CTH motif. That is an important component of the second complex. If we overexpress this motif, we can say that the signal transduction is negatively regulated. Yes. So based on all the results, we can say that if the receptor bind strigolactone and the strigolactone is hydrolyzed, then the receptor undergo a conformational change. Then it form a complex with the full-length D3 and the E3 ligase. Then it can trigger the degradation of D53 to activate the signal perception. If the complex was bound by another state of D3, that is to directly interact the sitting edge motif of D3, in this state, the receptor cannot function to activate the signal transduction. So we want to know what's the mechanism to terminate the signal perception, because based on our results, we can say that the strigolactone is bound covalently to the receptor. That is to say, once it can bind, it cannot leave. Then how to terminate the signal perception? We found that the receptor can undergo degradation and ubiquitination, and this process is dependent on the interaction with the F-box D3. And interestingly, we found that the receptor have an unknown N-terminal disordered region. This region is required for its degradation. If we delete this region, the receptor cannot degrade it. However, we found that this receptor without the N-terminal regions can show enhanced ubiquitination. So this is another, this is hard to understand because the protein is ubiquitinated very strong, but it cannot degrade. We found that that is a new mechanism behind this because the N-terminal region is important for the direct interaction of the receptor to the 26S proteasome. If the N-terminal region is delayed, it can interact with D3, so it can ubiquitinate it, but it cannot enter the proteasome, so it cannot degrade it. This is a new mechanism in plants. Then we found that the N-terminal region have another function, is that it can undergo phosphorylation, and the phosphorylation can negatively regulate to repress the ubiquitination of the protein. That is to say, the N-terminal region is important also for the regulation of the termination process. Then this is the activation and the termination region of the whole strigolactone perception process. What's the meaning of this process? We want to know whether this process is responsive to environmental signals. Importantly, we found that the low nitrogen signal can promote the D14 phosphorylation and to stabilize the receptor. And this process is important for the Taylor response to low nitrogen. As we all know that under high nitrogen conditions, the Taylor number is high, and under low nitrogen conditions, the Taylor number is low. This change is important for the grain yield, and that's why the farmers always use more and more nitrogen, because they think that use more nitrogen can promote the Taylor and can got high yield. But in the D14 mutants, although the mutant form many Taylors, and the Taylor number do not respond to low nitrogen. That is to say, D14 is the K proteins to mediate this process. And this mechanism is dependent on the phosphorylation and N-terminal, because if we mutate the phosphorylation or the N-terminal, the Taylor response to nitrogen is defective. Importantly, we found that there is a material. If we change the phosphorylation state of D14 that is to block the phosphorylation process, then the Taylor number of this plant do not change, even we reduce the nitrogen fertilizer by 15%. So this strategy is important for crop breeding, because we can perform the precise editing and to achieve the high yield and less fertilizer. So we summarize the whole story is that we know how does the strigolactone perception is activated, and we know how does it terminated, and we know that low nitrogen can regulate these two process, and this mechanism is important for the tolerant response to nitrogen. And after that, we want to know, after strigolactone perception, it needs to transduction. Then what's the downstream mechanism? Because although the hormone have lots of functions to plants, but the responsive genes is very, very few. People cannot identify effective responsive genes to explain the phenotype. We found that the key things is the chemical, because people use the chemical called RAC-DR24 to treat the plants, but this chemical is a mixture of two DR24 isomers, and these two isomers can triggered both strigolactone signaling and carotene signaling, and these two signaling pathways can interact with each other. So we produced by a new molecule called GR2440. These isomers have specific structures that can directly specifically bind the receptor to trigger the signaling transduction. And using these compounds, we identified hundreds of genes, and these genes can explain the important function of this hormone. For example, the shoot branching phenotype, the leaf elongation phenotype, and the isocyanine biosynthesis phenotype. Then we want to know how does the signal perception link to the responsive genes, that is the repressor proteins. Because in the classical hormone signaling pathways, including the auxin, jasmonic acid, gibberellin, and the strigolactone pathway, people think that the transcription repressor is the key component, and this repressor protein must bind with transcription factors like RF proteins, and to repress gene expression through repress the function of transcription factor. That is the classical idea. But we found that in the strigolactone signaling pathway, the repressor protein SMAX678 can directly bind the DNA, and it can function directly as a transcription factor. That is to say, it itself is the transcription factor. So we name it as a dual functional repressor proteins. It can function both as a transcription factor and the transcription repressors. These two functions can explain its role in the signaling homeostasis and also its function to regulate the development. Recently, we found that this mechanism is conserved in rice because the D15 protein in rice is the repressor protein. It can directly bind its own promoter to repress its own expression, and it can also directly bind OSPAL genes to regulate the rice blast resistance. And then we want to know whether is there any new mechanism yeah, the answer is yes. We found that the SMAX protein, that is the repressor protein, it can mediate the crosstalk of carotene and strigolactone signaling. And more importantly, this protein can function through directly bind and protect other proteins. For example, it can protect PIF from degradation by the 5B protein, so it can mediate the crosstalk with red light signaling. That is to say, the repressor protein can function without binding with the TPL, the classical pathways, and it have non-transcriptional functions. And then we want to know another important function of the hormone, that is to function as the molecular in the rhizosphere. And for example, this hormone is important for the striga seed and orobunchi seed to recognize the host plant, then to germinate. And this is because this is a very devastating disease in the world and cost billions of dollars every year. So we focus on the orobunchi resistant because this is an important disease in China. We use the tomato population to evaluate the orobunchi phenotypes in the field, and then we do the GWAS analysis and identify key genes responsible for the orobunchi resistant. We found that the genes, in fact, it encodes the strigolactone transporter. The transporter is mainly localized in the root epidermis, and it transmembrane protein. It mediates the exclusion of strigolactone from root to the rhizosphere. If we block the function of this transporter, that is to generate genome editing plants. And we can see that the mutant plants have very good phenotype. It can reduce the orobunchi parasitism less than 20%, and it can significantly increase the fruit yield in the field by more than 13%. That is to say, just manipulating one gene can realize the high resistance and less yield loss. And this mechanism is conserved in both tomato and sorghum, because we also collaborate with another lab. They work on the sorghum, found that the ABCG, another ABCG protein in sorghum is important for the parasitism with striga. And the last story is about the root microbiota, because whether rhizosphere bacteria can regulate tellurine and its molecular mechanism is a mystery, is unknown previously. We found that the root microbiota can secrete a small molecule called cyclo. The cyclo can directly bind to the strigolactone receptor of rice plants and to trigger the strigolactone signaling to inhibit the tiller development in rice. So this is a very interesting story, and to tell us that in the rhizosphere, the bacteria can directly interact with the plants and regulate the important agricultural traits. So that's all. And I want to thank the group member of my lab, and also Professor Jiayang Li is the team leader of our big team. And also I have many collaborators, and thank for the grants. So thank you all.
Thank you, Dr. Wang, for your insightful presentation. And the floor is now open for questions. Our committee members, please feel free to unmute yourself. Okay. The rest house, please. Yeah.
Yeah, I hope you can understand me. So I'm traveling and I'm sitting somewhere with a lot of people. Can you hear me, or is that okay?
Yeah. Yeah. Okay.
Oh,
Yes.
okay. So being great work. So very exciting, and you already somehow answered my questions that I had in between by showing data. Just for curiosity, so the strigolactone receptor complex, is it localized in the nucleus directly to degrade the repressors, or do you also find it in the cytoplasm?
I can't follow your last sentence. Sorry.
I was just curious about the localization of the strigolactone receptor complex. Is it directly active in the
localization.
nucleus?
Oh, yes.
Yes.
Yes, I know that. Localization, yes. We do that, but yeah, another story. The localization, it have dual localization. Yes, both in the cytoplasm and also the nuclear, and it can transfer. Yeah. So in the cytosol, people do not know the function of the receptor in the cytoplasm, but we found that it is important for the strigolactone hydrolase and to can regulate the endogenous level of the hormones, in fact. Yes. And so the function of the receptor outside the nuclear is very important and unknown topic. I think it is a frontier, really the frontier of this area. Yeah.
Yeah, thank you.
Yeah, thank you. Thank you.
Thank you for the questions. And Professor Frank House, please.
Thank you very much for this fascinating talk. I found it really great that when you mutated this transporter of strigolactone in tomato and sorghum, if I got it right, you
Yes.
then really no longer have the problem with the parasitic plants, which is
Yes.
good news. But I guess there must also be a dark side to this because plants don't just make such a transporter
Yes.
so that the parasitic plant can infect them, right? And so what
Yes.
do you think is the penalty for not having this transporter anyway? What is the negative side?
Oh, I do not say the detail of this story. In fact, in tomato, there are at least two transporters responsible for the exclusion of this hormone. We found that only the ABCD45 have the good result because it is not required for the normal development of the fruit. That is to say, it is not so important for the normal development because another transporter can also transport the strigolactone to the upground and to regulate the development. But it can, in this figure, you can see that we just see that they are reduced parasitism, but not no parasitism. If we completely block the transport or the biosynthesis, the plant growth is very bad. And also another thing is AM fungi. We also evaluate the influence of the AM fungi symbiosis of the mutant. Yeah,
Yes.
we found that the symbiosis is reduced, but yeah, it's reduced, but the reduced about I think reduced about 14%, but it do not influence the plant growth because we use many fertilizers in the field.
Yes, yes,
So
yes.
it's not have negative effects, the reduction. So that's a balance. So in this model, we think that's a balance. Yeah.
Yes.
Yes.
So
Thank
obviously,
you.
if in intensive agriculture, if we continue to use fertilizer, which we're trying to diminish, right? This is a
Yes.
good idea. But then the important question is going to be how well does it work if one uses less intensive agriculture with reduction of the use of fertilizers, for example, would then the positive and the negative, the balance between the positive effects and the negative effects be a little bit different? I guess that's the question.
That's right. So another frontier, I think, we are working now is that we cannot only depend on the plants. We need chemicals. We need chemicals that specifically influence the striga or orobanche, but do not influence the host plant. Then we can use the chemicals to solve the problem. Yes.
Very good. Thank you very much.
Thank you. Thank you.
Dr. Wang, please.
Yeah, just short question. So being great talk.
Yeah.
So I just wonder why less than 100 genes are induced by the strigolactone. It seems very few genes.
We identified hundreds of genes.
Yeah, but compared to auxin.
Yeah,
auxin
in fact, in rice you can got thousands of genes. Yeah.
But why? Whether you can see the
It's
difference.
very few. Not as many as other hormones, maybe.
Yeah.
Yeah. Because this in the biosynthesis and signaling process of this hormone, I mean strigolactone, there are lots of negative feedback. In every step, negative feedback. So just like the perception process, it can on within 3 minutes, then 2-3 hours it's off. So I think maybe this is the reason that why there is not so many responsive genes.
Yeah.
Yes.
Yeah. Okay. Thank you.
Thank you.
Okay, thank you for the thoughtful questions, and thank you again, Dr. Wang.
Oh,
Okay.
okay.
Thank
you.
Okay. Our fourth speaker in this session is Dr. Ruan Knight from Harvard University. His research centers on the chemistry of plant-derived molecules, from hormone biosynthesis to neuroactive alkaloids, uncovering novel enzymes and pathways with potential applications in medicine and agriculture. He will be sharing his work titled Novel Chemistry and Enzymes in Plant Metabolism. Dr. Knight, the floor is yours.
All right. Thank you so much for that introduction, and hopefully you can hear me okay. Well, yeah, so thank you again, in particular, for the organizers for providing me with this opportunity to share with you all some of the research that I've been doing over the past few years and that my lab has been doing since we started a few years back. And to begin, I want to take a step back and show you a picture that I took outside of my apartment while I was a postdoc. This is a borage seedling that was growing outside of my apartment. And the reason that I like to show this picture is that I think it really nicely demonstrates some of the challenges that plants face sort of in their everyday lives. And so this borage seedling was unfortunate enough to germinate out of a crack in the sidewalk, so not really the most ideal place to begin your life if you're a plant. And I hope you can see that basically on every single leaf, you can see little holes or little places where some pest or pathogen has been chewing on the plant, trying to eat it. And yet, despite these challenges, this plant has been able to thrive in this relatively hostile environment. And one of the major reasons that plants are able to respond to and thrive in these different types of environments that can be very challenging is because of their ability to produce a really huge number of small molecule chemicals that they can use to engage with the environment and fine-tune their own growth, development, and defense. And I think as a lab, that's what we're really motivated by, is the ability of plants to use molecules to grow and to defend themselves, to acquire nutrients, and to even interact with other organisms in a beneficial way, for example, pollinators or microbes in the soil. And we're really fascinated by all of the different chemicals that plants can use to accomplish these processes to thrive under these different circumstances. And as humans, we've long recognized the beneficial properties of these molecules from plants for our own benefit. And I think one of the most exciting places where this has occurred is within human medicine. And this is something that humans have been doing for thousands of years. Plants have provided us with some of our original medicines, some famous examples of medicinal plants I'm showing you here. And this isn't simply something of the past. We, of course, now know that the beneficial property of these medicinal plants is due to their production of very specific molecules that are able to interact with proteins in our cells and exert some kind of activity that's beneficial within the context of disease treatment. And the molecules that I'm showing you here are, of course, still clinically relevant. They're all currently FDA-approved pharmaceuticals and are still used in the clinic. And I don't really want to focus on the individual chemistry of these different molecules here, but I hope what you can appreciate is that these molecules have a really wide range of different structures. There's a lot of chemical diversity in the plant kingdom. It's estimated that there might be millions of unique molecules produced in plants. And I hope you can also appreciate that these molecules are very complex. And what we're really excited about in my lab is trying to understand how plants can achieve the biosynthesis of such complex molecules. And so really, we're trying to answer a very fundamental question in my lab, a very general question, and that is, both for plant biology as well as for us within, for example, medicine. And as we think about this in my lab, we really break this down into two major questions related to this. The first of these is relatively straightforward. What lists and biosynthetic pathways required to build these molecules? And so in essence, how do plants take simple primary metabolic building blocks and through the use of enzyme catalysts, convert these into these specialized natural products that allow plants to thrive in different ecosystems or which we can use as medicines? And in a practical sense, if we can elucidate these biosynthetic pathways in the underlying genes, we can then begin to think about how we could engineer these pathways in other organisms, whether it's microbial or other plants, to acquire useful molecules. But for my lab, I think what we're really most motivated by is the potential for discovering new enzymes and new chemistry that hasn't been observed before in nature. As I mentioned previously, there's millions of molecules in the plant kingdom, and we've really just scratched the surface in our understanding of how these molecules are made. And so we think that by exploring new pathways, new molecules, we're likely to find new chemistry that hasn't yet been discovered. The second question that we really want to try to answer is a little bit more complicated than this. And in particular, what we want to try to understand is how is this chemistry, these biosynthetic pathways, actually being organized in a multicellular organism like a plant? And I think a nice way to illustrate this is with a molecule that we're actually working on in my lab. So there's this molecule lobenaline, which is a neurotoxin produced in Lobelia species. And this molecule, as it turns out, is not produced everywhere in the plant at all times. It actually really only accumulates in this latex that you can observe when you cut or wound the plant. And very recently, we've been able to generate preliminary results that suggest that this isn't even where lobenaline is being made. It seems like it's actually being made in a different part of the plant and perhaps being transported into this latex. And this is something that's been continually observed within plant metabolism, is that you'll often have these storage cells or storage locations, in this case, latex cells, but this might be physically separated from the actual biosynthetic cells that make the molecule of interest. And you might actually have different populations of cells that are involved in biosynthesis, where you might have some chemical reactions occur in one cell population, transport an intermediate to another cell population, and then so on and so forth until your molecule actually ends up in the final destination. And of course, this organization of metabolism can occur within cells as well, where you can actually have subcellular compartmentalization of different enzymes and chemical reactions. And so collectively, as a lab, we're trying to understand not just how plants build molecules at a chemical level, but also how this chemical is actually organized in a plant. And we think that understanding both of these is really key for building a comprehensive understanding of how plants synthesize these complex molecules in nature. So these are our major questions, but you might be asking yourself, well, how do we actually uncover these complex plant biosynthetic pathways? Well, in my lab, we've settled on a very effective strategy that actually relies upon this conditional biosynthesis of molecules that I mentioned previously with lobenaline. And what I mean by this is that these specialized molecules aren't produced everywhere all the time in the plant, but often in a specific tissue type, at a particular developmental stage, or perhaps under a particular environmental stress. You could think of a caterpillar chewing on a leaf, inducing the production of a defense-related molecule. And so if the molecule is only being made in certain places at certain times, you'd expect that the biosynthetic genes that make the molecule are only going to be expressed where and when that molecule is being made. And it's a very simple idea, but it actually turns out to be quite powerful. And so we spent a decent amount of time in my lab to first try to determine what molecule is actually being made in the plant. And there's many different ways of doing this. You could imagine screening different tissue types or different conditions until you can actually find the site where your molecule is actually being made. And once you have this information, you can then pair it with transcriptomic profiling to try to associate gene expression with the actual production of your molecule. And we can do this relatively quickly and affordably these days using RNA sequencing of different tissues in our plant. And then finally, we're quite interested in actually characterizing the function of these enzymes that we identified through co-expression analysis. In our lab, we've been able to do this very rapidly by using what's sort of a common system now for studying plant biosynthesis, specifically agrobacterium-mediated transient expression in tobacco, which I'll talk about in a little bit more detail in a few slides. All right, so this is the general framework that my lab uses to try to understand plant biosynthesis. And as a lab, we've been using this pipeline to study many different complex alkaloids that are produced in plants. And many of these molecules have medicinal properties or neuroactive properties. But today, I really just want to walk you through one story that I think nicely highlights these different concepts that I've highlighted in my introduction. And specifically, I want to focus on this molecule, huperzine A, which has been explored as a potential treatment for dementia. So this is a project that I started as a postdoc, and it's been carried out quite nicely by the first graduate student actually to join my lab, Eric Fields. So huperzine A, or Hupe A, as I'll refer to it throughout my talk, is a major alkaloid produced in club mosses. So these are plants that belong to the Lycopodiaceae family. And it's one of many alkaloids produced in these plants that are collectively referred to as the lycopodium alkaloids. But hupei has garnered the most attention out of these alkaloids, largely because it's able to inhibit this enzyme, acetylcholinesterase, which is an important enzyme at the neural synapse in animals. And it happens to be a major target for many drugs that are used to treat the symptoms of Alzheimer's disease. And so hupei itself has been explored as a potential treatment for Alzheimer's disease, as well as a number of other neurological conditions. But at a more basic level, if we look more broadly in club mosses, what we observe is that there's over 500 unique lycopodium alkaloids that have been described. And if we look at the structures and functions of these molecules, we can see that they're quite diverse, and many of them have properties that would suggest they could be useful within medicine. But before we started working on this pathway, there's really not much known about how these molecules were actually made in the plant. And so this seemed like a really uncharted realm of plant metabolism that we could dig into to look for both new enzymes as well as new chemical transformations that perhaps had not yet been observed in the plant kingdom or in nature more broadly. And so to understand huperzine biosynthesis, the first thing that we wanted to do was understand where and when it's being made in the plant. And so to do this, we worked with a hupei-producing plant, Filmarius tetrasticus, which I'm showing you here. And I don't have time to go into all the details, but what we were able to show through a number of different metabolomic studies is that hupei is predominantly made in the new growth of the shoot. So this green tissue at the tip of the shoot here. And you can see that if you look at just hupei accumulation in the plant in different tissues, this is the tissue where it accumulates to the highest level, suggesting active biosynthesis. And so with this information in mind, we could then move forward with RNA sequencing of our plant, both biosynthetically active as well as inactive tissues, in order to have data sets for differential expression analyses. But as you might expect, after this transcriptomic analysis, what you're left with are tens of thousands of unique transcripts, and it becomes sort of a bioinformatics challenge of how do you actually identify the genes that are relevant to your pathway of interest or to our pathway of interest. And this brings me to a very important concept that we rely upon in biosynthetic pathway discovery, and that is this idea of biosynthetic or chemical logic. And what I mean by this is that if we have an understanding of the underlying chemistry that's occurring in our biosynthetic pathway, we can make reasonable predictions about the types of enzymes that are likely going to be involved in catalyzing that chemistry. And for huperzine A, we were fortunate that chemists had performed isotope tracer studies with club mosses going back to the 1970s. And these studies have provided really fundamental chemical insight into how these molecules might be made. And so, for example, these isotope tracer studies showed that the lycopodium alkaloids are ultimately derived from the amino acid lysine, as well as the fatty acid building block, malonyl-CoA. And I'm not going to go into all the different details here, but these isotope tracer studies allowed us to build a logical biosynthetic proposal for how these molecules are processed and put together and tailored to ultimately produce huperzine A itself. And I realize that on the surface, this is a lot of chemistry and it might not make a lot of sense. But what I want to emphasize here is that we can actually zoom in on specific parts of the pathway here to gain some insight about the types of genes that we should be looking for in our RNA-seq data set. So for example, in this first step in the pathway, it's predicted that lysine is decarboxylated to form this polyamine. Well, we know from previous work in the literature of enzymes that could do this, specifically lysine decarboxylase family proteins. And importantly, these are very easy to predict bioinformatically out of RNA-seq data. And so what we can do is look in our transcriptomic data set for lysine decarboxylase that's highly expressed at our active site of biosynthesis, and then use this as a bait gene and co-expression analysis to look for other genes that are likely involved in our biosynthetic pathway. And in this case, we might look for genes like copperamine oxidases, which we know from previous work are involved in oxidizing polyamines. We might look for polyketide synthases, which are known to act on malonyl-CoA as a substrate. And then for these downstream oxidative tailoring reactions, we can look for enzymes like cytochromes P450 or 2-oxyglutarate-dependent dioxygenases, the major oxidative enzymes that are typically involved in plant metabolism. And so if we use this exact strategy, if we look for a lysine decarboxylase or LDC gene that's highly expressed in our new growth tissue, and then correlate the expression of all genes in our transcriptome, what we find is that out of over 40,000 unique transcripts, within the top 25 or so correlating to our lysine decarboxylase, we can find many of the relevant enzymes that we were expecting. For example, copperamine oxidases, type 3 polyketide synthases, and a number of unique 2-oxyglutarate-dependent dioxygenases or 2-OGDs. And so it seems like this really simple co-expression analysis can lead us to the relevant genes that are likely involved in our pathway. And now we just need to be able to test them rapidly for function. And as I mentioned previously, the way that we do this is using this agrobacterium-mediated transgene expression system, which I'm guessing many of you are familiar with, but I'm going to walk through this quickly just to make sure everybody understands what we're doing here. But after we predict our candidate genes, as I explained to you, we can clone them out and then move them into an agrobacterium strain library, where each agrobacterium strain contains one gene construct of interest. Agrobacterium, of course, being a plant pathogen, we could use this to infect and transform our plant host. And in our case, we use Nicotiana benthamiana, a relative of tobacco. And this then expresses our proteins of interest that we can then test for enzyme function. And the really wonderful thing about the system is that we're not limited to testing just one enzyme at a time. We can actually do combinatorial testing where we added many different Agrobacterium strains containing multiple gene constructs to test for many enzymes simultaneously, or to build biosynthetic pathways as we discover new enzymatic genes. In terms of enzyme activity, we often rely on substrate from the tobacco plant that's natively present, but we can also add in substrates if they're not natively present, just as you would add in a substrate to an in vitro reaction in a test tube. And then after several days of incubation, we can extract our metabolites and perform either targeted or untargeted metabolomics to look for consumption of substrate or the production of any new molecules that might be relevant to our pathway of interest. And so to start, one place we could begin was to focus on the downstream part of the pathway. And we were able to do this in large part because a collaborator of ours, Dr. Yunyi Lo from the University of Malaya, had isolated an alkaloid from club mosses, this molecule flabelladine, that to us looked a lot like a potential precursor to huperzine A. And Dr. Lo was kind enough to provide us with some of this molecule flabelladine that we could use as a substrate. And so again, the idea here is we can take this molecule and infiltrate it into tobacco leaves that are expressing our oxidase gene candidates to see if we get any activity. And so if we added flabelladine to a control that's expressing GFP, we don't see any consumption of this molecule. However, when we add in one of our 2-OGD enzymes, what we're calling 2-OGD4 here, we get consumption of flabelladine and production of this new molecule where we have this addition of a carbonyl group. We can then add in an additional 2-OGD enzyme, get consumption of the second molecule, and then produce a new molecule where we have the installation of this double bond that I'm showing you here. And I'm not going to go through all the details here step by step, but using the same strategy of testing highly correlating genes, we were able to walk our way forward and show that flabelladine does indeed serve as a precursor to huperzine A, and that we had the minimal set of enzymes to convert this molecule into hupe. At the same time, we were able to jump into the early part of the biosynthetic pathway, and we were able to confirm that the lysine decarboxylase, copperamine oxidase, and type 3 polyketide synthases we had identified do indeed act as predicted to convert lysine and malonyl-CoA into these two key precursors to the lycopodium alkaloid. And you can see here from LCMS chromatogram traces that we get production of these molecules in comparison to a standard here. And using the same strategy, we were able to identify additional enzymes that co-express with our biosynthetic genes to move our pathway forward. For example, a dehydrogenase reductase, an acetyltransferase, a cytochrome P450, and very excitingly, a pair of enzymes that act together to catalyze this very unique reaction to form this tricyclic scaffold that I'm showing you here. And I don't really want to emphasize much of the chemistry on this slide, but what I want to draw your attention to is this scaffold forming reaction, because what we had actually found here was that a pair of carbonic anhydrase-like enzymes were acting together to catalyze this scaffold formation. And when we found this, we were very intrigued and excited because if you look in the literature, the only activity that's ever been attributed to carbonic anhydrase family proteins is this interconversion of carbon dioxide and bicarbonate. And of course, this is a very important reaction for basically all kingdoms of life. It's involved in things like CO2 solubilization, concentrating, and pH control. But to our knowledge, this is the first example of this enzyme family acting directly in natural product biosynthesis. And so we were really excited to look into these proteins in more detail and try to understand, well, how are they different from a canonical carbonic anhydrase? Well, if we look into the structure of a canonical carbonic anhydrase, the human COT2 protein, which is very well studied, what we see in the active site is this histidine triad that coordinates a zinc ion. And this chemical moiety is actually critical and essential for this canonical carbonic anhydrase activity. Well, if we look at alpha-folded structures of our cal proteins that we had identified from club mosses and look at the same residues in the active site, what we can see is that this histidine triad is either partially mutated or completely mutated, suggesting not only that these cals no longer bind zinc, but that they likely catalyze their reaction in a fundamentally different manner. And if we look more broadly at the active site residues present in the canonical active sites, we have this histidine binding, zinc binding histidine triad, as well as these other active site residues. What we find is that if we look across all kingdoms of life, these residues are extremely highly conserved in carbonic anhydrases. So this includes animals, bacteria, algae, fungi, and even plants. We don't really see any changes to these residues in the canonical enzymes. And so it was really striking to us to see that these changes occurred in our cal proteins. And the first thing that we thought was perhaps we could use these mutations as a guide to find other neofunctionalized carbonic anhydrase-like proteins throughout the plant kingdom. And the first place that we wanted to look, of course, was in Lycopodium alkaloid biosynthesis. And right away, we were able to identify 3 additional cal proteins that had these striking mutations to the active site, suggesting that they may have neofunctionalized enzyme activity. And importantly, all 3 of these proteins co-expressed with the huperzine A pathway, suggesting that they were involved in Lycopodium alkaloid biosynthesis. And I don't have time to go into all the details, but what we've been able to show subsequently is that 2 of these new cal proteins, cal3 and cal4, act together in some context to catalyze this reaction that I'm showing you here. And you might be wondering, what is the relevance of this reaction? What is this molecule S4PAA? Well, as it turns out, this molecule is actually one of the 2 substrates that's used by the other 2 carbonic anhydrous-like proteins, cal1 and cal2, in this tricyclic scaffold formation. And so it seems like collectively these cal proteins are acting together somehow to enable the scaffold formation. And so we wanted to learn a little bit more about what was happening here. Well, one of the things that we noticed with these proteins is that all 4 of the cals we had identified had these predicted N-terminal signal peptides, suggesting that they were not cytosolic, but rather might actually be secreted outside of the cell into the extracellular apoplast. And so we actually wanted to confirm this. And so what we could do was tag our cal proteins with histidine, 6S histidine tags, extract apoplast selectively from tobacco leaves, expressing our proteins, and then perform Western blots. And what we found is that in comparison to a cytosolic control protein, lysine decarboxylase, which doesn't accumulate in the apoplast, we could actually see robust levels of the cal proteins in the apoplast extract. And I don't have cal4 shown here, but more recently we've been able to show this for cal4 as well. And so this result suggests that the cals are actually secreted to the apoplast. And exciting for us because, as I mentioned, the rest of the enzymes in this pathway seem to be cytosolic. And so it seems like there's maybe this physical separation of different pathway components. And so this allows us to build an initial model to think about how organization might actually be occurring in Lycopodium alkaloid biosynthesis, where we have these early biosynthetic transformations from lysine and malonyl-CoA happening in the cytosol, some sort of mysterious transport mechanism that we don't fully understand, and then the cal proteins acting extracellularly in the apoplast to catalyze the formation of this tricyclic scaffold that I'm showing you here. And I think this is a very interesting model, but one thing that you might notice from a previous slide that I mentioned is that we've already characterized the downstream enzymes converting phleboladine into huperzine A, and all of these proteins are in fact expected to be localized to the cytosol. So we sort of have this problem potentially of this very highly polar tricyclic intermediate being stuck in the apoplast. And we were curious if transport was perhaps an essential part of this pathway, if there was an essential role for transport within huperzine A biosynthesis. And to cut a long story short, that's exactly what we ended up finding. And so what I'm showing you here is this same tricyclic scaffold shown in a slightly different orientation. And what we had found is that a specific reductase was involved in the next step of this pathway, but it only worked if we co-expressed a transporter protein, this peptide transporter like PTRL1 that we identified through co-expression analysis. And what this looks like in terms of LCMS data is that we can produce this tricyclic scaffold with our early pathway genes. If we add in the transporter, nothing happens. And if we add in the reductase by itself, we also get no activity. It's only upon the co-expression of the transporter with the reductase that we get depletion of our tricyclic scaffold and the production of this new molecule. And once we were able to find this, we were then able to find the final enzymes in our pathway, cytochrome P450, and an acetyltransferase that allowed us to produce phleboladine itself. And so this then provides a even more clear picture of what might be happening at an organizational level, where we probably have production of this tricyclic scaffold in the apoplast, and then subsequent transport through the specific transporter protein into the cytosol, where the final enzymes can then take over to produce phleboladine, and then ultimately huperzine A. And we still have a little bit of work to do in terms of actually characterizing the biochemistry of this transporter. But so far, all of our data points towards this being a likely model for what's actually happening in terms of biosynthesis in these plants. And I hope this really emphasizes, at least at a small scale, how important these concepts of metabolic organization are in terms of understanding plant biosynthesis. But at this point, we felt like we had identified the minimum set of genes that were necessary to convert primary metabolites from the plant into huperzine A. And so the final experiment that we wanted to perform was, well, can we take all of these genes and move them into the tobacco plant and actually demonstrate de novo biosynthesis of huperzine A? And to cut a long story short, this worked. So we could actually take all of these genes, move them into tobacco, and show that we could actually produce this club molecule. And though we're not producing incredibly high levels of this molecule, I think it's a very nice proof of concept that we can very quickly go from pathway discovery to pathway engineering in just a matter of years and really accelerate this discovery process. And so with that, I want to wrap up my talk and leave you all with a couple of major takeaways. Hopefully, if you don't remember anything else from my talk, you'll remember these points. So first, I hope that I've been able to show you effectively that we really have a powerful strategy for identifying biosynthetic pathways of plants. We can go very quickly from a medicinal plant with a known medicinal compound, and through this combination of metabolomics, transcriptomics, and rapid testing in a plant host, discover complete biosynthetic pathways and then engineer them in a model plant in lab. I hope I've also been able to convince you that organization is a critical component of plant biosynthesis. And obviously this idea of transport has been brought up in previous talks today. But in our case, the discovery of this transporter was essential for us being able to engineer the production of this pathway. And I think this is something that's going to become increasingly true as we study plant metabolism, and is going to be a really key component of any efforts to engineer these pathways in other organisms. And then finally, I hope that I've been able to convince you that plants are really rich with novel chemistry. And one of the examples that I provided to you today is this exciting example of carbonic anhydrase-like proteins neofunctionalizing to catalyze new reactions. But I think as we continue to explore different molecules, different pathways, we're likely going to find other new enzymes and new chemical transformations that will sort of set a precedent for the chemistry that's possible in nature and in the plant kingdom. All right, so with that, I'd like to end my talk. I want to make sure to acknowledge everybody in my lab, in particular, Eric Fields, who led this work most recently on Huperzine biosynthesis. My previous lab at Stanford, the Satley lab, where I initiated this work, Dr. Yunyi Lo, who provided us with critical standards for this process. Of course, I'd like to thank all funding sources, both past and present. I'd like to thank all of you for your time, and I'd be happy to take any questions you may have.
Thank you, Dr. Knight, for a really stimulating talk. Question time, and our committee members, please feel free to unmute yourselves. Okay, Francis, please, Professor Francis, please.
Thank you very much for this beautiful talk. If I got this right, you have early steps of the biosynthetic pathway happening in the cell. Then you have this intermediate step happening in the apoplasm, and then the final steps go back into the cell. And so I was wondering if it's possible that actually the whole thing happens in two different cells. You know, you could imagine the first cell, and then you have an intermediate, which is then extracellular, and then it's picked up in another one. I'm not sure what that would mean exactly, this separation, but is this something that you think is possible, or?
Actually something that we're trying to follow up on right now. So the short answer is we don't know the answer to that question right now. If I had to guess, I would expect that they're actually the early part of the pathway and the late part of the pathway is occurring in different cell types. And I think there's a couple of reasons that that might be happening. So first of all, I didn't really touch on this, but those early biosynthetic transformations produce intermediates that are extremely reactive. And so if we put all the enzymes together in tobacco, I sort of gloss over the details, but we get tons of other non-specific products because we're mixing molecules that I think are not normally mixing in the club moss. So I think separating cell types is a very effective way to keep reactive intermediates away from each other. A second point to this is, I didn't really touch on this either, but there's many different lycopodium alkaloids in addition to huperzine A, and there's branch points in the pathway that happen right around the place where the carbonic anhydrase-like proteins are working. And so I think you could have different cell populations downstream that are actually involved in producing distinct molecules. And so you could actually separate whether you want to make huperzine A or some other alkaloid in time and space. And so we're following up on this with single nuclei sequencing to see if we can learn something about where these genes are actually expressed. But I hope that I'll be able to share something about this in the coming years, or hopefully months.
Thank you very much.
Thank you. Any other questions? We still have time. We still have 2 minutes, please. Maybe we still have time for one question. Okay.
Yeah, maybe just for curiosity. So initially, in the beginning, you mentioned lactifiers, and I also liked a lot the idea of Christian that maybe, yeah, in the end, some of these metabolites end up in lactifier cells, and being generated in or initial steps from surrounding cells. So I'm wondering, what is known about the membrane of lactifier cells? Are they full of transporters for secondary metabolites?
You're acting in specifically in club mosses or more broadly? So I mean I think in the case of club mosses, there's really not much known about this role of transport, and this has been the role of transport in specialized metabolism has been well studied in some other systems, for example, opium poppy and Madagascar periwinkle for production of some other alkaloids. But in terms of what's actually happening in the club moss, we really don't have any idea. There hasn't been any effort so far to look into how this is actually happening in the native plant.
Yeah, thank you.
Okay. Thank you, everyone. And once again, thank you, Dr. Knight.
Thank you.
Okay. Our final speaker today is Dr. Tatsuya Nobili from TSL UK. His research pioneers single-cell and spatial transcriptomics to uncover the cellular complexity of plant immunity responses and deepen our understanding of plant microbe interactions. His talk is titled Decoding the Molecular and Cellular Architecture of Plant Microbe Interactions. Dr. Nobili, the stage is yours.
Well, thank you, Ram, for the introduction. And I also want to thank all the organizers and committee members for this opportunity to share with you our work. So I've been really fascinated by how plants interact with microbes, especially bacteria. And today I'm going to tell you a few stories where new discoveries or new biological insights were made possible by developing and using new technologies. So I'm going to start with a short story from my PhD work, which was about 6-7 years ago. Now, plant-bacteria interaction is always a two-way process, and we know many things about how bacteria influence plant responses, especially immune responses, either inducing or suppressing immune responses using different molecules such as MAMPs and effectors. But relatively speaking, much less is known about the other way, how plants influence bacterial responses. And one big question back then and still now is that how does plant immunity actually stop bacterial pathogens? What's the influence that eventually stops bacteria? To address this big question, we developed a method that allows us to directly analyze bacterial responses in plants under strong immune activation. We call this method as implanted bacteriomics, where bacterial cells are physically isolated from the infected plants. And then we can extract RNAs and proteins from the same sample to do transcriptome and proteome analysis. So it's a multi-omic analysis. So without this enrichment, typically you get 99.9% plant information and you're not able to analyze bacterial responses. So now with this method, I'm going to show you one example using the transcriptome data. So we analyze bacterial transcriptome in a variety of Arabidopsis mutants of defense pathways, showing different levels of immunity. And we saw this remarkable dynamic changes in bacterial transcriptomics. And I'm still struck by this figure showing that bacterial transcriptome patterns at an early stage of infection in plants, this x-axis, correlated very strongly with bacterial growth at much later time point. So this means that early bacterial responses that are heavily influenced by immune responses of plants are tightly regulated or tightly linked, with bacterial virulence. So by leveraging this method, combined with more classical plant omics, we were able to understand the interactions of the molecular networks of both plants and bacteria. For example, we revealed that plant immunity suppresses bacterial ion homeostasis pathway, which is partially causal for pathogen growth suppression. So at least for me, this method development really changed how I understand, how I see plant microbe interactions. And since then, I'm really motivated to develop new methods to understand new biology. And related to this topic, there are recently very exciting studies. For example, this paper this year from Miao et al. Identified plant molecules, metabolite, that directly target bacterial virulence mechanism to disassemble the virulence machinery to stop bacterial growth. So with all these omics data, I think we are getting closer to understand and answer the question of how plant immunity actually stops pathogen growth. So I encourage you to check this space in the next few years. Okay, so this was my introductory short story. But now let's step back a little bit from talking about each molecule, so genes and proteins, and let's think about how cells, how plant and bacterial cells might interact with each other. So this is a confocal image of Arabidopsis leaf infected by bacterial pathogen with GFP. What you see here is a heterogeneous pathogen colonization in the plant. And when you think about plant responses, because plants don't have any specialized and mobile immune cells like we do, each cell has to decide what to do immune responses, or there are many other fundamental functions such as photosynthesis or cell division, nutrient allocation, and so on. The plant cells are inherently multitasking. And you could imagine what happens might be something like this, right? Depending on the cell type or relationship with the pathogen cells, each plant cell might respond differently. But we really didn't know how exactly this kind of cellular heterogeneity might look like, because traditionally we mostly focused on analyzing the whole tissue, kind of averaging things out. So this is the current state, and our challenge is to bring this state to this state. And to do so, we use single nucleus multi-omics, which of course we didn't invent that technology, but we have done a lot of optimization of this technology to our model system. Again, Arabidopsis leaf infected by bacterial pathogens. We used different strains, both immune suppressive strains or activating strains, and many different time points, those key time points. The idea here is to diversify the situations that plants might experience so we can capture a lot of different cell populations. We develop a quick nuclei isolation protocol, and then we use single nucleus multi-omics, which allowed us to analyze both transcriptome and epigenome, in this case, open chromatin regions, from the same cells and thousands of cells at the same time in a single experiment. And I guess you're familiar with open chromatin region, but those open chromatin region can be bound by transcription factors and cofactors to regulate genes. So we could understand potential regulatory mechanisms. So that's why we use this method. Now, here's the overview of the single cell data we obtained. We sequenced a little over 65,000 cells from all these different conditions. And this allowed us to identify those major cell types, but also we can further subcluster those clusters to identify much more granular cell populations sharing similar transcriptome patterns. And systematically, we identified, classified over 400 cell populations or subclusters. And here I'm showing a couple dozens of famous immune genes. And what you can see immediately here is how diverse those immune gene expression is across these cell populations. So this means that upon pathogen attack, there are many variety of immune cell populations that express different immune genes in different combinations, which is something quite new and for us, and it's quite exciting finding. Also, we can leverage the multi-ohm data, which allowed us to directly link chromatin accessibility to gene expression at the single cell or cell population level. And in this way, we identified thousands of genes that are significantly correlated with nearby accessible chromatin region in a cell type specific way or cell population specific way. And now our lab is currently following up on the actual regulatory mechanisms of cell type specific gene expression. For example, certain transcription factor might bind to this region, to this gene. So a short takeaway from this part is immune activation creates diverse cell populations with coordinated transcriptome and epigenome changes. Now, the single cell multi-ohm data is really data rich, but we are not quite this state yet, right? Because we're still missing the critical spatial information because all the nuclei are released at the beginning before sequencing. So we have no idea about where those cells came from. So that's the next big knowledge gap. To fill this gap, we used another new technology called spatial transcriptomics. We specifically used a method called MERFISH. MERFISH is a massively multiplex single molecule imaging technique that can visualize hundreds of different genes at the same time on the tissue section at the single molecule resolution. So we applied this technique to exactly the same kind of tissue, a Arabidopsis leaf infected by the pathogen. We cut this longitudinal section supported by my former colleague, Travis Lee. And we selected 500 genes for this MERFISH experiment. As you can see, we did exactly the same time course experiment, including mock and pathogen-infected samples. This is immune activating pathogen. And here I'm only showing about 12 genes out of 500 genes, but you can already see this massive transcriptional reprogramming upon pathogen attack with some interesting spatial patterns. And what we did was we integrated this image-based data with all the sequence-based data of single-cell multiome analysis that I showed you earlier to model the spatial temporal dynamics of immune responses at the single-cell resolution with full transcriptome and epigenome resolution. So what you see here is upon pathogen attack, you see the emergence of those immune-active hotspots that are growing over time and then changing their states. So now we have this, for me, this is kind of a treasure map. And we wanted to really identify some novel biological mechanism of plant immunity. So what we did was we focused on what's there at the center of those immune-active hotspots. So we zoomed in and we found something quite interesting. So what we found was two fundamentally different cell populations. One is we call them as primer cells for primary immune responder cells. We believe they are in direct contact with the pathogen. The other cell state includes cells surrounding primer cells. We call them as bystander cells. So bystander cells may not directly see the pathogen, but can still activate the immune responses because primer cells can tell bystander cells. So this is quite simple in concept, but our contribution is that we provided rich molecular information that can distinguish these two cell populations. For example, this is the spatial transcriptome data. You can see the yellow molecules, BON3 gene. It seems to be enriched in the primer cell population, whereas LD1, FMO1, more classic immune genes are enriched in the bystander cell population. And completely independent single-cell RNA-seq experiments also supported this, so BON3 and these two genes are separately expressed. So now our lab is following up on the actual function of different cell states. We identified dozens of many other cell states in addition to these two, and also the regulatory mechanisms. For example, primer cells are quite interesting. It's very rare cell state and full of genes with unknown function, probably missed because of it's a rare abundance of primer cells. For instance, we identified a transcription factor called GT3A, which is clearly enriched in the primer cell population based on single-cell RNA-seq data. Again, completely independent spatial transcriptomics showed that GT3A showed very similar spatial expression as BON3, which is a primer cell marker, as I showed you in a previous slide. Also, the single-cell attack-seq data, the epigenome profiling, showed that in the primer cell cluster, the GT3A binding sites are more open genome-wide, so they are ready to be bound by GT3A. So all in all, the data suggests that GT3A might be an important transcription factor in this primer cell. And we do have data showing that GT3A indeed contributes to immune responses. So the message here is that by integrating single nucleus multi-omics and spatial transcriptomics, we can identify a lot of cell populations, including the very, very rare cell population, such as primer cells. And we can further understand potential mechanisms regulating these cell states. So here's a simplified model. We identified, in addition to dozens of cell populations, we are excited by those two populations, primer and bystander states, which I believe represents cell autonomous and non-cell autonomous immune responses. They seem to be regulated by distinct transcription factors and also regulatory elements controlling different genes, likely having different functions. Also importantly, there should be mechanisms to communicate between primer and bystander cells, which is completely unknown currently. So it's quite exciting open questions that we are addressing currently. So the take home here is that even though plants don't have specialized immune cells like we do, each cell upon pathogen infection can flexibly turn into diverse cell population or immune cell states and potentially coordinate with each other to deploy this multicellular immune network to protect the tissue. And I'd like to take this opportunity to propose that immune cell states like primer and bystander cells are fundamental building blocks of the plant immune system. And here I'd like to briefly explain possible factors that can influence on the diversity of immune cell states. So the one thing is pre-existing immune potential. So this means that even before pathogen attack, different cells might express different immune receptors in different levels or combinations. And also the downstream molecular network configuration may be different. And therefore, even when same pathogen comes, those cells might turn into different states. Also, there is the cell autonomous and non-cell autonomous immune responses that can create this kind of gradation pattern. Also, pathogens move over time and they might decide to do different things at different places, obviously affecting cellular responses of plants. And in reality, these things happen at the same time, making things really, really complicated. And to really disentangle this complexity, we need single cell and spatial resolution and omics level molecular information. And I think it's a very exciting time because of the emerging new technologies. I recently wrote a review here summarizing emerging single cell and spatial omics technologies, especially focusing on those that have not been applied to plant biology, let alone plant-microbe interaction studies. That includes different modalities, including epigenome to transcriptome, translatome, proteome, and metabolome, and so on. And also the combination of them, so-called multi-omics, single cell or spatial multi-omics. So if you're interested in the recent developments, I hope this review will give you some useful information. And for our lab, the immediate next step, after having this single cell multiome or RNA-seq atlas and spatial transcriptome atlas of merfish, which is two-dimensional, is to get the 3D information with pathogen and to plant together, because pathogen information have been lost in those two data sets we generated. And unfortunately, none of the commercially available platforms allow us to do this. So we developed our own tool, which is the FightMap. So FightMap is a multiplex single cell and 3D spatial gene expression analysis method that can work in thick tissue, even whole mount plant tissue. So as a proof of concept, we used the Arabidopsis root tips, which worked quite well. So here we marked some of the known or predicted cell type specific genes in the root, which beautifully showed different cell populations. So currently, FightMap can target up to 50 genes, but could be 100 genes with more optimizations. And the protocols are relatively straightforward. Any molecular biology lab with confocal microscope can do it. And it's quite low cost. And also importantly, it's transgene free. So you don't have to make new reporter lines, which is very time consuming and labor intensive. And this means that you can directly tap into all the genetic resources, such as mutant collections or natural variations. And I hope FightMap can help bridge this spatial biology with genetics to really go into the mechanisms. And our lab has been helping many researchers across the globe to implement phytamap in their own laboratory, and with a lot of success, and it's been quite rewarding to see phytamap being used by many people, addressing many other questions that goes beyond our immediate questions. So if you're interested in this type of technique, feel free to reach out. I'm happy to support. And we're obviously implementing phytamap in this leaf pathogen interaction model to directly follow up the studies I showed you earlier. But today, please let me stick with this root system, because we have another very exciting line of research using root. And our question is how roots interact with the surrounding microbiome in the soil. And the root microbiome interactions are arguably even more complicated and heterogeneous interactions compared to, let's say, plant versus a single pathogen. So we really want to resolve the plant microbe interactions at single cell and spatial resolution. And today I'd like to share with you some preliminary data, which is, to me, a technical breakthrough for us to achieve this goal. So we applied phytamap to visualize bacterial colonization or multiple bacterial colonization in planta. So what you see here is the root of Arabidopsis colonized by 4 different commensal bacteria, originally isolated from wild Arabidopsis root. And you can see phytamap could visualize their colonization. So different strains show different niche preference. So for example, Agrobacterium occupies this main root cavity, and the bacillus seem to prefer the root hairs, and also you see some other patches here and there. Again, this is preliminary data contributed by many of lab members, such as CU Son, Sean Pai, and Hazel Surtees. But we are extremely excited by this, and we're obviously excited to expand this to many other bacterial strains, of course, with rigorous benchmarking. And our dream is to spatially map every single cells in the microbiome at the single strain resolution to exactly know who is sitting where with whom. And at the same time, we visualize plant gene expression responding to many different environmental conditions, including microbiome, but also nutrient availability and so on. And linking those two information to the actual phenotype to understand how cellular interactions of plants and microbiome are translated into the organism level or even holobiome level traits. And I believe this can open up a lot of new opportunities in plant microbiome research. So towards this, we're trying to develop a dual phytamap that targets both plants and bacteria at the same time. So I'm almost done. So today I introduced 5 technologies that we contributed. Here's a quick recap, especially focusing on those that we are using in our lab actively. They have different features for molecular depths and also spatial resolution. Sequencing-based method is unbiased, but the more targeted phytamap has 3D. So integrating all these methods and further advancing new method will allow us to access information that haven't been accessible. And that's also essential to addressing key biological questions. This is the last slide, but what we really want to do using those new methods is that we want to bridge our understanding of the molecular basis and cellular basis of plant microbe interactions. What I mean by this is, in my view, the major discoveries in the past, let's say, decades in plant microbe interaction or plant immunity research have been in this space, the molecular basis, including the discovery of resistance genes, which turn out to be immune receptors in ARs, and their protein structures, downstream signaling, hormone networks, or chemical diversity. These are very exciting molecular bases. And many of the recent discoveries were actually made by many of the speakers in this symposium, which is quite amazing. However, we still lack the foundational understanding of the cellular basis of plant microbe interactions. So what kind of cells there are, And I propose that plant immune cell states may be useful framework to understand this. Also, we cannot ignore the bacteria or microbial part, as I introduced at the very beginning. And also by focusing on certain cell states, we may be able to discover novel function of genes and proteins as implicated by Ryan's talk or also Tony's talk during this symposium. So I think it's a very exciting time to bridge those two with new technologies. And I hope we as a community can collaborate and using interdisciplinary and creative approaches to tackle these questions. So I'm always happy to discuss what we can do together. All right, so with this, I want to thank all the people I had the privilege to work together, especially my former mentors, Ken Tsuda for PhD and Joe Ecker for my postdoc, and all the amazing lab members and TSL colleagues. And thank you very much for your attention, and happy to take any questions.
Thank you, Dr. Nobili, for wrapping up our session with such an inspiring talk. We now welcome any final questions from the committee. Please feel free to unmute yourself. Dr. Xiaofeng Cui, please.
Hello, Tetsuya, very nice talk. So one question I want to know how you inoculate the Abdopsis plant leaf by infiltration or by spray the bacteria?
Yeah, thank you. That's an important question. So we infiltrate the bacteria at very low dose where fewer bacteria will be there relative to plant cells to create this heterogeneous situation. But thank you for the question.
Okay, so if you by infiltration, but bacteria infect plants and the natural physiology condition, maybe not by infiltration, maybe by other ways, like through the stomata or through the water pore in the leaf. So your discovery of the primary cells and the bystander cells in the leaf can also, is be identified under the natural infection condition.
Oh yeah, thank you very much for this important question. So our initial study used very simplified model, which is infiltration, which bypasses stomatoimmunity that adds additional complexity. But now using phytomap, or also we are making a lot of reporter lines as well, marking the primary cell bystander cells. And once we have those lines, we can actually test natural condition or even different variety of pathogen to see whether primary cell bystander cells are conserved across variety of pathogen resistance or not. So that's our next goal.
So another question is, I fully agree with you that new technology can be used to for new discoveries. So you use bacteria as a model system. So I want to know, do you have any comments about if you study the other types of the pathogen, like the necrotrophic pathogen, vascular infected pathogen, can this technology you introduced today can be used to study this pathogen interaction with the plant?
Yes, absolutely. So we have some collaboration project targeting some filamentous pathogens, including fungi or mycete. Our event, and ultimate goal is to really make this technology versatile to any kind of interactions. And in nature, plants interact with a variety of different pathogens or non-pathogen, even symbionts at the same time. So we hope to visualize everything together. That's kind of the ultimate goal.
Okay, thank you.
Okay, thank
Thank
you.
you.
And Professor Frank Hauser, please.
Thank you very much for this beautiful talk. I have a quick technical question. And I think in your final slide, you said you can monitor up to 20,000 genes, but I thought that single nuclei, one of its limitation compared to single cell is that the sequencing depth is actually not quite as good. So how many did you actually really manage to get?
Oh, is that so 20,000? Oh, so this is so per cell or per nucleus, you get much fewer, typically
Okay.
700 to 1,000. But what we do is by clustering the similar cell population together, we will get the equivalent coverage to the bulk RNA-seq eventually, or bulk ATAC-seq. So still, I hope the technology will improve to, for the sensitivity. But what we can do now is to sequence more cells to cluster them to get sort of cell state level coverage, but like a full deep coverage of molecules.
But then in a sense, you defeat the purpose of single cell, right? Because you basically have to put together many different cells so that eventually you get all of you see what I mean? It's
Yes. It also touches upon a little bit philosophical question, because eventually every single cell is unique. But to really do molecular biology with replicate, we need to somehow classify cells into certain populations. That's why I'm proposing as one possible framework, this immune cell state concept, which enables us to really deeply analyze and actually in a genetically tractable way. So until we have the perfect technology that can measure every single molecule in the cell real time, I guess we have to just try to do the second best option. And my bet is, the cell state classification might help us get there sooner.
Thank you very much. Beautiful.